Hey everyone, welcome to The Explainer. I am absolutely thrilled you're here today because we're looking at something literally revolutionary. We're unpacking Dr. Jean-Claude Perez's groundbreaking biomathematical discoveries, specifically his work, the Codon Octave Handbook, Unlocking the Musical Architecture of Life. Buckle up, because we're about to completely rethink the very blueprint of human existence.
For decades, we've been handed this very specific story about biology. It makes you ask, is human DNA really just a random, chaotic string of letters? Is it just this messy biological soup that kind of fell together by chance over millions of years? We usually picture these 3 billion characters piled up with zero overarching structure, right? Just sitting there waiting for a ribosome to grab a tiny piece of it. But what if that textbook view is just completely incomplete? What if it's missing the actual design?
Okay, let's dive into this. Traditionally, we see the genome as this chaotic mess, but look at Perez's biomathematical view instead. When you actually put on a mathematical lens, those 3 billion characters stop looking like random noise. They start looking like a self-designed, highly structured numerical whole. It's actually scored with the absolute mathematical precision of a Bach fugue. We're talking about an elegant, biomathematical symphony.
Part 1. The Hidden Genomic Symphony, Defining the Foundation.
Now, to actually hear this hidden genomic symphony, we need to zoom all the way in and define our basic notes. And before we go any further, we have to make a crucial distinction here, separating triplets from codons. Triplets are your 3-base non-overlapping DNA words, like AGT. These act as the master template. Codons, on the flip side, are the mRNA transcripts, the copies of those triplets that later get translated into amino acids. By looking at these billions of unbroken triplets as a massive unified population, this high-precision meta-architecture actually starts to reveal itself.
And this brilliantly highlights the core mathematical constraint of life. Biology builds 20 amino acids out of just 4 DNA bases. To pull that off, it groups them by 3, which spits out exactly 64 possible combinations. Not 63, not 65, exactly 64. That is not just some biological fluke, it's a rigid mathematical parameter that literally defines the boundaries of our entire genetic language.
Moving on to Part 2, Unlocking the 8x8 Matrix, the Numerical Filter.
So, we've got our 64 combinations. But what happens if we stop looking at them one at a time and instead treat the entire population of billions of triplets as one giant unified whole? Well, the standard biological lens treats the genetic code like a simple dictionary for protein synthesis, right? Just mapping 64 codons to 20 amino acids to crank out organic matter. But Perez tells us to use a mathematical frequency lens. Instead of just caring about what the code makes, this lens looks at a matrix of frequency. It works like a numerical filter, exposing the exact proportions and frequencies of every single triplet across the entire genome.
And guys, it is genuinely mind-blowing when you see the result. When you take those 64 triplets and sort them into a descending scale of frequency, meaning from the absolute most common down to the absolute rarest, they physically reshape themselves. They don't just spit out a list, they arrange into a perfect crystalline 8x8 grid. 8 rows, 8 columns. We call these rows octaves.
Let's take a look at the very top row, octave 1. This is the foundation. It holds the absolute master peaks of frequency in the human genome: TTT, TCT, TAT, TGT, ATT, ACT, AAT, and AGT. These aren't just random groupings of letters. These are the heaviest, most universally dominant anchors of our entire genetic sequence.
And the thing is, this isn't just some flat, boring grid. These 8 octaves physically cluster into thematic groups to guarantee massive structural stability. Master octaves 1 and 3 team up to form the high-frequency cluster. They are the foundational resonance. Then octaves 2, 4, 5, and 7 group into the medium-frequency cluster, which gives us the massive redundancy we need to protect genetic info. Finally, the absolute rarest triplets drop into octaves 6 and 8, the low-frequency cluster, providing this really subtle but super necessary contrast.
Now, what's really interesting about this is in Part 3, 3 constants of life, the numbers governing DNA.
Because these octave clusters, they don't just float around willy-nilly. They are locked into place by rigorous mathematical laws. The entire genome is balanced by 3 specific genomic numbers acting as mathematical attractors. First up is the number 1, which stands for the strict parity balance between complementary DNA strands. Then we have the number 2, representing this perfect 32-32 partition, where the top half of the codon table is used exactly twice as often as the bottom half. And finally, phi, the golden ratio of 1.618, the universal proportion of harmony.
Which leads us to part 4, symmetries and piece symbols, the math in action. The absolutely crucial thing to grasp here is that these constants aren't just abstract ideas on a chalkboard. They force deep, verifiable symmetry right into our DNA. Let's actually walk through what Perez calls the mirror codon algorithm to see this in action. It's a gorgeous expression of this symmetry. Take a triplet, let's say TCG. Step 1, you reverse it to GCT. Step 2, you swap each base with its biological complement, so G becomes C becomes G, T becomes A. The final result in step 3, CGA. TCG and CGA are mathematical mirror twins, and the genome perfectly balances their populations across its entire length. No way that's an accident, right?
And watch how this mathematical constraint translates into pure geometry. If you divide the entire codon population into 4 quartiles based on these numerical constraints, the exact ratios 2, 3/2, and 1/2 literally draw a perfectly proportioned piece symbol. I know, it sounds crazy, but this isn't some quirky design coincidence. It is a direct geometric manifestation of the strict mathematical constraints keeping your genetic data mathematically balanced.
All right, part 5, the golden ratio balance, form versus substance. This brings us to the absolute climax of Perez's findings. All of these octaves, all these wild symmetries, they exist to serve one overarching architectural purpose, bringing form and substance into perfect harmony using the golden ratio. Remember those 8 octaves we talked about? Well, they divide perfectly into a dual structure. 5 of them act as the biological substance of the genome, while 3 of them act as the mathematical form. And get this, when you divide the 5 substance octaves by the 3 form octaves, the result is exactly 1.618, phi, the golden ratio. The exact same ratio that dictates the spiral of a galaxy or the curve of a nautilus shell is literally governing the balance of your DNA. Just think about these numbers for a second. 13 and 144. These are famous Fibonacci numbers, intimately tied to the golden ratio. In the human genome, the precise ratio between the total 64 triplet population and the absolute most frequent triplet pair is exactly 13. And when you expand that analysis to all reading frames, meaning the different ways the cell actually reads the sequence, it hits the resonant Fibonacci number of 144 perfectly. The resonance is just undeniable.
Now, you might be sitting there wondering, okay, so what? Why does all this math actually matter? The ultimate takeaway here is that this mathematical elegance is an absolute physical necessity. The human DNA double helix is massively long and surprisingly heavy at a molecular level. If it weren't for this absolute mathematical symmetry perfectly balancing the literal atomic weight of the strands, the human genome would be physically unstable. It would literally collapse.
To really put that into perspective, look at this incredible number, 1.00000045. Because of the golden ratio and those octave constraints we just walked through, the atomic mass of strand 1 is mathematically tuned against the atomic mass of strand 2 to a precision of nearly 1/millionth. 1/millionth. That is the level of absolute engineering we're looking at inside every single one of your cells.
As we wrap up this explainer, I really want to reflect on this quote from the ancient Pythagorean principle. The beginning, which is 1, is the middle, which is 2, of the whole, which is phi. Thousands of years ago, philosophers inherently understood these universal laws of proportion. And today, biomathematics is proving that the computable numerical order of the human genome follows those exact same ancient harmonic laws. Which leaves us with a pretty mind-expanding question to ponder. If the very blueprint of human life isn't just a biological accident, but rather a mathematically perfected symphony, what other hidden songs are playing in the universe around us, just waiting for us to find the right mathematical lens to finally hear them? Thank you so much for joining me on this explainer. Keep learning, keep questioning, and I'll catch you next time.